So hello everybody, welcome to today's Ento Live webinar, Flying Squids, their life story and relationships with each other. We're really lucky to have Fernando zooming in from Spain today. The reason that this talk is in the program is I received a newsletter from, I think it was a Linnean Society, and it had a link to this paper in it. And I was just fascinated by the idea of flying squid. And I knew straight away that I had to get a talk on this, partly for the program, but mainly for myself. It's something that I'm really looking forward to learning more about. And I'm going to hand you over to Fernando now, and he's going to amaze us with the world of flying squid. So Fernando, over to you.
Hello to everybody. Thank you very much for listening. Thank you very much, Karen, for the presentation. So we are going to talk today about these flying squids. That is one of my favorite topics. And we are going to start, with this question. Do you think that flying squid fly? I think you will see a pool there with several options. Kieran, can you confirm that this,
Yeah, sorry, it should be there now. It should, you caught me off guard there, Fernando, too quick. So you've got a poll in front of you there asking if flying squids fly, and you've got a number of answers. Those potential answers are no, they don't fly. Yes, they spend around 90% of their life flying. Yes, they jet propulse themselves and glide, or yes, but only on their vacations. So yeah, I think Unfortunately, I'm surprised that all of the answers are getting a vote there. Right, I think we've got most of the people have voted. I'm going to end that poll there. So if you haven't got your answer, you've got like 2 seconds. Right. Okay. So share the results, Fernando. What we've got is 84% of people believe that they do fly and they jet propulse themselves and glide. We've got a small proportion, 11% saying they don't, and a few people have went for what I see as are the trick answers of only on vacation and 90% of their life flying. I don't know the answer. I'm looking forward to hearing it. If it is 90% of their life flying, I'm going to be particularly wowed, Fernando.
Okay, well, we can move, and this is the answer here. Yes, they can glide over the water. When there is a predator below the water and they want to avoid this predator, they can just use the water that is inside the mantle of the squid to create a jet propulsion that propels there over the water for 30 meters or so. So they can just do that, and they don't spend 90% of their lives doing that. So that's something very punctual. It's very difficult to see. And there is here this beautiful photo for Okano Tetsuya that you can find in his Twitter that shows how they work, how they fly. So we are going to keep talking about these squids, but first I want you to present you a little bit more about cephalopods. So cephalopods are mollusks, so they are the closest relatives to the snails, clams, and other mollusks. So they are quite different, you know. They are very intelligent, and they are so fascinating in their biology that maybe it's some surprising that they are very close relative to snails and animals like this, but that's what they are. And regarding the cephalopods that we can find, the extant cephalopods, we have this group here, isolated from the remaining ones that are the nautilus. They have a very biological aspects, very different from the remaining cephalopods. And most of the diversity of current cephalopods are the coleoid cephalopods here with this black background. So we have the octopus, we have the cuttlefish, and several groups of squids. We have the coastal squids, the main groups, and then the oligopsid squids, that are the oceanic squids. Also, we have a lot of biodiversity of fossils. These 2 photos came from the Natural History Museum of London. And here you can see a giant ammonite. And here you see another one that is a mosasaurus. You know, it's a reptile that lived in the Cretaceous, I think, not sure about that, sorry. But the thing is, if you zoom on this image, you will find a lot of these ammonites that fossilize over this body. And that's because, the ammonites were very diverse and very abundant in the Mesozoic. So during the time this animal, this Mosasaurus, was fossilizing, there is a lot of ammonites that just fall dead over the body and fossilized with it. So that's a pretty cool thing. So the thing is, cephalopods were very more diverse in the past, but they are still a very important group in marine environments. And it's also pretty diverse, as you see in these photos. So for now, we are going to just focus in these oceanic squids, in the flying squids, that it is a single family within these animals. It's the family of Mastrephidae, which has several names, especially in Spain, is because there is a lot of culinary tradition of these animals. We eat a lot of them, so we call them potas, potones, cananas, or potarros. Potarros is my favorite because it's from Asturias, my hometown. But in English, the name is flying squids. Here there is another photo of flying squids just trying to avoid a predator under the water, but they should be careful because they have another one above it. So they can be eaten during the flight. That's another amazing thing that can happen. They are characterized by this T-shaped mantle funnel-looking apparatus. Sorry about the technical name, but it's the name it has. So if you open a flying squid, you will see in the place where the head ends and starts the mantle, you will see in the funnel cartilage this inverted T, and also in the mantle. So if they have this, it's going to be a flying squid for sure. And they are also characterized by the morphology of the paralarvae, the first stage after they hatch from the egg. So this paralarvae has a name that is also very technical. It's the Rhynchoteutium paralarvae. And it's very morphologically easy to identify because they have this proboscis that is enigmatic. We don't know what they use it for. And it's formed by the two tentacles of the squid. They have 8 arms here. And then they have the 2 tentacles. And when they hatch, they have it fused in a single proboscis. And they also have the buccal filamental papilla around the mouth, this small papilla here, if you can see it, that is also characteristic from this family. No other squids have them. About the general aspects, we can talk about the size, for instance, and these species can be very small, like this Yaloteutis pelagica. This is a full-grown female, I think. And then we have here this cobalt squid that has 25 kilograms of weight. The record of this species, I think, is 60 kilograms of weight. So they can have all this variation in size, as you can see here, with the roller as a scale, and here with a human being as a scale. And where they live? Well, they live mainly in marine oceanic waters. So many of these species live in pelagic environments very far away from the coast. So they don't have any relationship with the sea bottom during their lives. And there are others that live in the slope, and there are others that live near the continental shelf. So like these two. There are some of them that even can have resting behaviors over the sea bottom, especially during the day. So, but there are others that never touch the bottom in their lives. And what do they eat? Well, they eat, they are predators, and they eat mainly small fishes like lentilfishes and krill, but sometimes they can get larger prey, as in this image of a Humboldt squid, like the one that you can see in the other photo. And I also want to tell you that these species are heavily fished and they're heavily consumed in several countries. This is from Madrid. That is very typical to get a squid sandwich that usually is it's made from flying squids because they are the cheapest. Yeah, that's the thing. But I encourage you to try it. It's a good thing to eat. So regarding the fisheries, this family of squids represent the most heavily fished family among all cephalopods together, because 50% of the total cephalopod declared landings in the world belong to this family. And the mainly fished species is this, the Humboldt squid or the jumbo squid that occurs in the Pacific, and it's fished by the artisanal and industrial fisheries of Mexico, Peru, Ecuador, and Chile. So in the whole world, you can get products made by this squid. For instance, this came from a market here in Spain, and we don't have Humboldt squid in Spain, but it's one of the main trade squids. So people have been saying that this species, the Humboldt squid, is the most studied squid of the century because it has a lot of interest for fisheries and also very interesting physiology. But here we are just talking about fisheries. The other main fish species is the Argentine squid Argentine squid, sorry. Here you have a photo. In the Mediterranean, we have 3 species that are fished by the trawler fishery mainly, that are Ilex coindieti, the first one, Todaropsis eglane, and Todarodes sagittatus. And then we have another species that is very important in Japan, that they fish them and keep them alive. And in some restaurants, you can find this pool in which you can fish it, and they will prepare sashimi from them. That photo is from Hakodate in the island of Hokkaido in Japan. Regarding the taxonomy, and this is the how we classify scientists, how the scientists classify these animals, we can say that there is currently 25 accepted species of the family Omasteridae, and there is some controversy reminding how many subfamilies this family is. That means how we arrange these species within larger clusters. Based on morphology, there is a proposal that classify them in 3 different subfamilies. But when people start to use the DNA to find different ways these squids evolve, we find out that these 3 subfamilies diagram didn't fit what we found. And some people, myself among them, are using now 5 subfamilies for this family of squids, but there is still some problems, and in my case, I am trying to fix them. We will see later on. So we are going to talk now about the life history of these flying squids. In this slide, there is the full life cycle of these squids represented. So we can start with the mating. Here we have the smaller male mating with the female, then the female release the eggs release the hatchlings eat whatever they eat in the plankton, and then we have the juveniles. The juveniles are the ones that can glide over the water during this phase of their life, and then they get too big to be able to be propulsed over the water by the jet propulsion of the funnel. So they stop doing that anymore. So let's talk about that. As I told you, we can place the beginning of this life cycle with the mating of the male and the female, in which the male transfers the sperm packets that are known as spermatophores to the female. These spermatophores create a reaction and start to releasing the sperm on the female. During this part of the life of the spermatophore, we call them spermatangium or spermatangia in plural. So the male uses a very modified arm for transfer these spermatophores to the female. And where he put these spermatophores or spermatangia, they do in different parts depending of the subfamily of these flying squids. In the case of the genus Ilex, that is the family Ilicine, the males put several groups of spermatangia that look like this one. So every of these is an spermatangium, so it came from a single spermatophore. So here it should be around almost 200 spermatophores put it together. They put it near the opening of the oviducts. So when the oocytes came from the ovarium and came to the external exit of the oviduct, they get fertilized by these spermatophores that are being released. But in the remaining flying squids, in the 21 accepted species that are not from the genus Ilex, the process is quite different. In this case, the male transfer the spermatangia to the oral area of the female. The spermatangium start releasing this sperm, and somehow this sperm gets inside the seminal receptacles. The seminal receptacles are organs from the female that store the sperm for many weeks or months. And they can store sperm from different males, so the final egg mass will be will have multi-paternity, and that's a good thing for the squids because they can have higher genetic diversity within their hatchlings, and that's a good thing for being more adapted under different changes in the environment and environment conditions. So here are the spermatangia, 3 of them, and here we have the seminal receptacles. After this, and then the females produce the oocytes in the ovary. These oocytes came from the oviducts, and then they go outside through the oviduct opening. And we have these 2 glands, the nidamental glands, here labeled as an NG, that create a mucus covering that would protect the eggs. Here we have a depiction of how these egg masses are created, and this is a photo of an egg mass in the wild. So they are quite large, and they float over the pycnocline of the water. So they are in the first 25 meters depth, just floating, and the embryo would develop in there. So just here a summary: male and female mate. Here there is a photo of a mating between 2 humboldt squid. Then the male insert the spermatangia around the mouth of the female. This spermatangia in the surface of the oral membrane release the sperm. This sperm goes by active swimming towards the seminal receptacles, enter inside, and get stored there for weeks or months. But when the female wants to release the egg mass, well, just the process I just mentioned, when the female wants to release the eggs, these spermatophores will be active, and they will release by the muscle action of the seminal receptacles, and they will, meet the oocytes and create the embryo. Here there is a photo of several spermatozoa heads going towards the direction to the oocyte, and here is how a fertilized egg looks like. So this is an egg that has met a spermatozoa, and now it's developing. This embryo development can be variable in length according to the temperature. Here there are some experiments we did in the lab, and they can hatch in 10 days at 17 degrees, but if you increase the temperature to 21 degrees, it will be just a week of embryonic development, and then you will have a fully developed paralarvae. When later on, the paralarvae just hatch and abandon this egg mass that is formed by mucus mainly, and then we have this ring-coated paralarvae I told you before, with the proboscis and the buccal filamental papilla. I will ask you again another question, and I will let you. I will like to know how many squids do you see in this image. So you will have some time now for answering.
Yeah, it's coming now. Bear with me. No, I don't want that poll back. Right, so you should see a poll launching now. It should be there already available for you. So yeah, so this question is about the image that's on the screen. All the same species, but that's the wrong one. Ignore that. No, end that poll. Sorry. That's not what I wanted to do. What it isn't a poll that's going to come up. What I want you to do is message in the chat. How many different individual squids do you see there? So yeah, drop a message in the chat, just a number. Can you see 1, 2, 3, 4, 18, 300? I don't know the answer. Right, we've got Diana has suggested 2.
Okay.
We've got Vicky suggesting 2, Hannah suggesting 2, Jen suggesting 2, Annika, a lot of people saying 2. Tony says 2, but this can't be true. Claire is going for 200.
Okay.
Lizzie knows it's a trick question, but she doesn't know what the answer is. I've got 170 from Terry, around 100, too large, but maybe about 100 baby. So I think on that note, do you want to let us know what the answer is, Fernando?
Yeah, for sure. So I saw that many of you answered there are 2 of them. Here we have a female that usually they are larger than the male. These 2 are fully grown specimens. So yeah, there are 2. But if we have a look in the corner, in the left down corner, sorry, you would see here another one. And this is the size of a hatchling. So squids in their lives grow from this size to this size. And that happens in a few months. This squid, in this case, is Todarodes sagittatus, and can live from 12 to 14 months. So just imagine how much they need to eat to grow that large size. So about the life cycle of these animals, they can have a maximum life cycle of 2.5 years, and that happens only in the jumbo squid, but usually they have a life of 6 to 12 months. So they grow a lot, as you can imagine. So they hatch with this size that is 1 to 2 millimeters of mantle length. The mantle length is the standard longitudinal measure that we take, that comes from here, from the edge of the head and the beginning of the mantle towards the apical tip of the mantle. So we have this early paralarvae that I described before. Then we have the advanced paralarvae, and they have changed a little bit because they start to develop a little more the arms, as you can see here in this image. They are longer and have more suckers. The proboscis starts to get stitch. That means that the tentacles start to break down by the base, and they will free themselves as 2 raptorial tentacles, and they lose the oral papilla. And when people open them, they will start to see prey inside their guts. And then they get the definite morphology that has in the adults, previously passing by the juvenile and the subadult stage. The tentacles get fully developed and they reach sexual maturity. So regarding the diet of these animals, cephalopods usually are predators, as this colossal squid in the cartoon. But we have 3 exceptions. As I told you before, the nautilus are very different to the remaining cephalopods. And among these differences that they mainly eat carrion. So they don't hunt as much as coleoid cephalopods do. But among the coleoids, we have the vampire squid that feeds mainly on detritus. And then we have the ram's horn squid, that is the spirula squid, that eat mainly detritus, but also hunt. And then we have the remaining squids that are just predators. So regarding the early paralarvae, I want you to ask you again something. Just remember what makes the flying squid paralarvae so special. So I mentioned twice, and maybe you can remember what I did. I think Karen should ask a new poll now.
Sorry, I've accidentally muted Fernando instead of unmuting myself. Yeah, the poll's just coming now. So this, I need to relaunch that. You get to vote again on the poll I accidentally launched earlier. So this is about paralarvae. And the question is, what makes a flying squid paralarvae so special? Is it that they already fly? Is it that they have a proboscis? Is it that they have filamental buccal papillae? Thanks for that one, Fernando. Is it that they can eat a tuna juvenile in 7 seconds, or is it all of the above? I know what I would guess, but I don't know. I'll let you know when people have put their voting. So about half of you voted. I'm going to give you just a couple more seconds. So if you haven't decided, get it in quick. Okay, I'm going to end that poll now, guys. Right, okay, so the most popular answer is that they have a proboscis, 47%. I can't eat a tuna in 7 seconds, surely. That's what puts me off all of the above. No, they don't already fly. Yeah, I'm going to agree. They have a proboscis. What's the answer, Fernando?
Yeah, the answer here is 2 things. They have the proboscis and they also have the filamental buccal papilla.
I only let them put 1. So anybody who put either of those, it's set to only allow 1 answer. You got it right.
Okay, yeah, that's the thing. So here we have the proboscis, as I told you before, the 2 tentacles fusing a single proboscis, the full filamental buccal papilla that is only found in this group of squids. And they also have something that is pretty interesting. They have a scarce development of the nervous system sensory organs, the digestive system, the respiratory system, the arms, and the beak. And this degree of development is equivalent of what we found in embryos of squids that had as predatory animals. And when people tried to find out what they were eating, just trying to recognize by visual assessment what was inside their guts, they found something very difficult to identify. They found like a mass that they weren't able. So we tried to solve this problem using the DNA of what it was inside the guts. But you need to keep in mind that these animals are very small. Here, the scale bar is 1 millimeter. So what we did was to process these squids using histology that creates a very thin portions of the squids. These are 7 micrometers of length. And we use laser capture microdissection, that is a methodology mainly used in biomedicine, to cut small portions of these histological sections and just keep them for further analysis. So what we did was to use this machine that you can use for select a portion of tissue. Here you will see a green line that is coming from the part I am selecting. Then you ask a laser to cut it following this green line. And then you just use the same laser to use a small, to point it in a small part of the slide. And then the part of the tissue you want will fall within a small tube. Here there are several of them, 3 of them, that only have the good content of the squid and none of the squid tissues. So that allows us to study the DNA of what it was inside the guts and not of the squid. And we also study larger squids that were far easiest to dissect and extract the good content. And then we extract the DNA and study what it was inside by the means of the DNA metabarcoding that gives you a read for each prey that is inside them, more or less. It's in a nutshell what we did. And what we found, most of the reads were from fungi. The second most frequent grab was the vascular plants, terrestrial plants. Then we have the animals, but half of the animals were about the half of them were terrestrial animals. And then we have other microorganisms like diatoms, freshwater algae, and other groups. So with this information, we try to infer what the paralarvae eat. And we ask ourselves, is this diet compatible with predation? If it will be a predator, we will find that marine zooplankton should be the major component of these threats. What we found, it was fungi, vascular plant, terrestrial animals, diatoms, and cyanobacteria that should be a minor component or just being absent. So we ask ourselves, can be an herbivore? And the answer here is that if they were herbivores, they would eat mainly marine algae, and the remaining components we found should be absent. So it's not also an herbivore. What we infer is that they are detritivores. So they eat particularly organic material that can have a marine origin, explaining the presence of marine organisms, and also a terrestrial origin, because many material that is inside the lands can go to the seas, towards the through the river and the wind and everything. So this material would be part of the detritivore food web, and during the rotting process would be enriched in the fungi. So that explains also the presence of fungi. Another source of particular organic material are the feces of zooplankton, and many of these zooplankton are herbivores, so that would explain the presence of diatoms. So we reached this conclusion based on what we found. So despite the adults being voracious predators, these flying squid paralarvae are detritivores. And it's very important because they can exploit a resource that is ubiquitous and accessible without competing with other flying squids for prey. And we think that this change from detritivory to predation is a unique strategy among predatory cephalopods. So that might be one of the reasons why this family is so abundant in the pelagic environments. I will talk now about something that is also very cool, that is the plasticity at the size of maturity. And I will speak again about the jumbo squid. Here there are two photos. And what you might not expect is that the two photos depict a mature female. So this 25 kilograms is a mature female, and this 250 grams female is also a mature female. And that's because there are three morphotypes that can be variable regarding the size at maturity of these females. And these three morphotypes has different latitudinal distribution. So in the equatorial part, there is hotter waters, and in hotter waters they develop quicker and within a shorter time frame. So you would have the small morphotype. In the extremes of the distribution range, like in Chile or in the Gulf of California, the waters are more cool, they are less hot, so they will grow for longer periods and they will grow more. So the larger morphotypes are in the extremes of the distribution of these species. And they also happen to change with the environmental conditions. Maybe you know the El Niño phenomenon that changed the patterns of wind in the Pacific area, and during the El Niño conditions, usually you have poor algae blooms. So the food webs in these environments during these seasons would be less abundant, and the squids stop growing so much. They grow for a shorter period and start to reproduce with a smaller size. So after El Niño in 2010, you see this change in the maximum size at maturity of the females. And this condition has been maintaining for several generations. Here you have a photo comparing the size of two mature females that were fished the same day in the Gulf of California. It was during El Niño conditions. So after that years, we only can find the small morphotype because the food webs in the area are less abundant. And people has been asking, it's the same species if they can be so large or so small. And they did molecular testing, and they found using the DNA that they are the same species. So that's a pretty cool thing these animals can do. They can decide if they want to grow for a longer period and be larger, or just stop growing and start producing eggs and just die off. They have also, these squids, very important ecological, they are very importantly ecologically very important, because they are important predators of small fauna like krill and lanternfishes, as already mentioned. They are also important threat for marine megafauna, as sperm whales, dolphins, and other mammals. And they perform large migrations, so they transfer energy from the feeding grounds to the spawning grounds, and so on. And after the spawning, they create massive mortalities, and the body of these squids sank towards the bottom of the sea, and then they feed the benthic communities in the bottom. So they, in the end, they are very important because they transfer large amounts of energy and matter from lower trophic levels in the marine trophic webs towards higher the trophic levels like the marine megafauna I mentioned. So now that we talk about the life history and the ecological importance of these squids, we are going to try to answer some evolutionary questions. And the first one I was interested in is to know what was the closest relative to flying squids. And here I use this depiction of a evolutionary tree that has been developed by Charles Darwin. I love this image, but we are going to do some evolutionary trees using the DNA for answering this question. So what I did for many years was to collect the squids of several families of oceanic squids, and that's not an easy thing because they live in very isolated environments, so I needed to go on ships and living there for weeks and months to try to collect these tissues for squids. In the end, I was able to get 22 out of the 25 oceanic squid families, not only by myself, also taking tissues from museums and other places, and other collaborators. And I studied the DNA to infer these relationships among these families of squids. So what I used was the mitochondrial DNA. So every of you are eukaryotic organisms. So you have cells that has a nucleus with the DNA, and then you have the power machineries of these cells that are the mitochondria. It's the ones that allows you to move your muscles and think and everything because they produce the energy you need. And these mitochondria also have DNA inside. And sorry, I don't know what happened, but it says that my function of sharing my screen it stopped.
No, we can still see, Fernando. Do you want to try going back a slide?
No, yeah, I see now. Sorry about that. So what I did, what we did was to use the DNA within this mitochondria to produce a strong phylogenetic tree that represent the evolutionary relationship within these squids. So I won't enter in this part of the tree that includes many families. I won't only speak about the squids that are related to the flying squids. So here in light blue, we have the family of Mastrefidae, the flying squids. And here in light green, we have the diamondback squid here in the photo labeled as B. That it's the closest relative. So the family of Mastrefidae and Thyssanoteutidae, the diamondback squids, are sister groups. And these two groups are the sister group of the family Cranchidae that are the glass squids. So we are going to talk about this cluster of three families that we label it as the superfamily Cranchioidea, using the stem name of the Cranchidae. And these are the Cranchioidea. We have the flying squids, you know a lot about them. I won't insist in their morphology. And then we have the diamondback squid that has some morphological similarities, but very few of them. And then we have the Cranchidae that has absolutely nothing to do with the flying squids and the diamondback squids. And also the Cranchidae can be very morphologically variable among them. We have the glass squids that are these transparent that can grow large, very large, or can be very small. These are the, the glass squids. And then we have the colossal squid, that is the heaviest squid on Earth. They only occur in Antarctic and subantarctic waters, and they have a very thick muscle layer that is not similar to the one that we found in other members of the family Cruncidae. So we have three very dissimilar species. So I want to ask you, do you think it is possible to describe a group of animals if they do not share morphological characteristics? And you can either say yes or no.
Yeah, so we'll just give you a couple of seconds to answer that poll. So yeah, just to repeat, is it possible to describe a group of animals if they do not share morphological characteristics? So I'm assuming there, Fernando, you mean to describe them as one group?
Exactly. That was my question. Sorry. Yeah.
Right. Okay. Right. I'm going to end that poll now. Right. So the results are in. Yes, 90%. No, 10%. Over to you, Fernando.
Okay, I see. So there is some people that think that you can't. The thing is, you actually can't. You actually can. And what we did was to use the structure of this mitochondrial DNA we sequenced. So here there is 4 examples of how the genes can be arranged in the mitogenome of one of these squids. And the last one is what we found in the superfamily Cruncioidea. So there is a gene that in the remaining squids is in this position here, where this arrow, this violet arrow I am pointing at now, the first one, usually the gene is in that position, but in all the Cruncioidea is in this other position. And here in this phylogenetic tree is represented the gene order of the mitogenomes, and as you can see here, all the Cruncioidea are exactly the same. So you can find this gene order in every Cruncioidea has been sequenced so far, and you can't find it in any other family of squids. So that's what we call an apomorphy, and it's what we use for describing a group. And that's what we did. We create this diagnosis, and if you look at the morphological part of the diagnosis, it's not very informative. So we have a small to massive adult size of Opsida. That means nothing, and with modifications in the funnel mantle looking apparatus, but these modifications are very different among them. And the remaining characters can be found in any other squid. But this gene order would also point towards a Cruncioidea. So in this case, we use the position of a single gene to use a diagnostic character for a particular group of squids. So that's something we did. And now we know which are the closest relatives to the flying squid. We are going to talk about the relationship among the flying squids itself. So as I told you before, people is transitioning from a 3 subfamily diagram within the or subgroups within the flying squids towards a 5 subfamilies taxonomy. That's the one I support. And this was based on previous work using some molecular data that were less powerful than the approach I am using, using the full genome of the mitochondria. So I try to answer these questions because this tree have several problems, especially regarding the position of the Ornithoteuthis. That is one of my favorite genera of squids, and also the genus Todaropsis is problematic. And we are going to see how it looks like, this phylogeny constructed using the full genomes of the mitochondria. But before trying to understand the results, you need to know what we want to find in phylogeny. Usually we want to find monophyletic groups. That means that you have a tree you cut it from one of these hypothetical ancestors, and you get all the descendants from this group. That is a monophyletic group, and that's what you want to get when you do phylogeny and taxonomic decisions based on that phylogeny and these evolutionary relationships. But it's quite often that some groups that you create are paraphyletic, and that means that you cut the tree at some hypothetical ancestor, and then you get many of the descendants, but not all. So here in light group, you will see how a paraphyletic group looks like. And that's what we found. We found that the genus Todarodes is paraphyletic. You see here, you have 2 species of Todarodes here, another 2 here within the genus Martalia, and then 2 more within the genus Nototodarus. So not all the not all the descendants from hypothetical ancestor are collected within this genus Todarodes. And the same happens with Nototodarus. If we cut the tree here, we have 3 species of Nototodarus and then 2 species of Todarodes within it. So we need to do some work here. And there is a missing species. Before going that, we found cryptid biodiversity. We found different species that were not sequenced before. So we need to describe these new species at some point. And as I was telling you, there is a species that I didn't have at the time I did this work, that are the genus Ornithoteuthis. But recently, I was in Twitter just looking what people were sharing, and I found a guy from Japan that was sharing one of these Ornithoteuthis squid. And I asked him, can I get a small piece of tissue for this squid? I need it for my phylogenetic studies. And he told me, sorry, I eat it already. So I asked him again, and he was so nice to send me some squids. So right now I have the squids in my lab, 3 of them, and I am already doing the sequencing. So very soon I will have also the genus Ornithoteuthis in my phylogenetic tree, and I think I will solve the phylogeny of flying squids. So I am very thankful for this guy. And one thing I want to mention, I was thinking he was a biologist because he has a deep knowledge on the morphology of these animals, and he was incredible collecting them, identifying them, and selecting tissues for me, but he is a fisherman. So it's something that I really, really appreciate. And well, we talked about which are the closer relatives of the flying squids, how are the relationship within the flying squid, that is an ongoing project, and then I am going to talk to you about flying squid cryptid biodiversity. And I will focus in this genus, in Omastrephes. Please keep this name in mind. But before I start to talk about Omastrephes, I am going to talk you to tell you what cryptid species biodiversity is. And with this clumsy diagram, in the left side of the slide, you have this vertical axis. That is the time. It comes from the past. Here you have the live ammonite drown, the time passing through, and then we have the ammonite fossil. Every ammonite in current times are dead, and they are just stone fossils. So here, this line, this discontinuous line, means the present. So we are going to think about an ancestral species that lives in a fire environment, for instance. And then these species start to diverge, and there is some reproductive barrier among populations of one place that still lives in the fire environment, and others that start living in the water environment. So their morphology changes, and then you have several morphological characters that arise to adapt to these changing environmental conditions, and you would be able to differentiate these related species just by the look of them. But let's imagine another scenario in which this ancestral species, we can label it like B in this case, just diverge, but they still live in a very similar environment, and still they have a reproductive barrier, so they speciate. So in that case, you can't use the morphological cues to identify these species. And that's when we talk about a cryptic biodiversity. There are species that you can't differentiate, but using their DNA. You can identify them using the morphology. So in the case of Omastrephis bartrami, that is the distribution that was depicted in the books back in the day, it's the second in size within the family of Omastrephida. The largest one is the Humboldt squid and the jumbo squid I told you before. This is the record of a flying squid. It was of the neon flying squid, the genus Omastrephis. It was 30 kilograms of squid and 1 meter of mantle length. And they are also fished in the North Pacific. And that's something important also for you, as you will see later. So when I saw to this map, the first thing I asked myself, it was, how it is possible that the North Atlantic and the Mediterranean populations would be able to reproduce with the ones that are in the South Atlantic and the Indian Ocean, and then these ones with the ones that are in the South Pacific and the North Pacific, because there is large gaps in distribution where there is no neon flying at all. So it was a weird distribution map for me, and I asked myself, is this a single species or there are more? So what we did, it was to collect neon flying squids, genus Omastrephis, from the whole world, and what we found, it's different groups. We have the group that occurs in North Atlantic and the Mediterranean, this group 1. Then we have another one that occurs here in Cabo Verde, Brazil, and the Indian Ocean. Then we have a third group that occurs in the South Pacific. And then we have the fourth group that occurs in the North Pacific. Remember, it's the place where there is a target fishery for these animals. And we use what we call a species delimitation analysis. That is some software package that, using a evolutionary tree, can infer how many species are there. And most of them were consistently drawing a 4 species, output. So, and also, if you see of this cholinesterase activity, it's the activity of enzyme that occurs in the brain, and they found differences between South Atlantic, North Pacific, and North Atlantic. And there is also differences in the morphology of the spermatophores, the ones from North Pacific and the ones from North Atlantic and the Mediterranean are very different to the ones from the remaining part of the world, and also difference in the size at maturity of the females. So there is a small morphological evidence that we have 4 species, and a strong molecular evidence that we have 4 species. Molecular evidence means that the DNA points out towards 4 species of the genus Omastrephis. And as I told you, there is no much morphological evidence of these 4 species, so we use specific positions of the DNA to label each species. So here in this position 334, we have a G in the one of group 1, that means guanine, and A, that means adenine, in the group 2, and then we have thymine in the remaining oceanic squids. So we can use these positions as an apomorphism for each species, as we did with the gene order of the Crunchoidea. When we don't have morphological support, we can still use their DNA and find characters that we can use to say, well, if it has a C here, a item in 306, it's group 1. So what we did, we label which oceanic barriers can create these differences among populations here. This Canary Current can be one of them, and then the North Equatorial Current can create a barrier between this group 1 and this group 2. And that means that we have here an allopatric speciation. That means that a larger population exists in the past, then an oceanic barrier occurs, that can be these currents, and they start to go apart from each other, have their evolutionary trends across different populations, and create 2 different species, in this case with a very discrete geographic pattern. So you here in the North Atlantic always have group 1 and you wouldn't have group 2. So now each species has a smaller distribution range that the previous Omastrephes bartrami distribution range I showed you before depicted. And that leads to a taxonomic revision. So we find a name for each of these species. So Omastrephes caroli occurs in the Mediterranean and North Atlantic. Omastrephes cylindasius occur here in the Gulf of Guinea and the Cabo Verde, the Brazil, South Atlantic, and the Indian Ocean. And then Omastrephes bartrami occur in the North Pacific, and Omastrephes brevimanus occur in the South Pacific. And this is important also for fishery assessment. As I told you before, there is a single target fishery for neon flying squids, and this occurs in the North Pacific over Omastrephes bartrami. That means that if we don't manage properly this population, and we just erase this animal from the North Pacific, we will just extinct any species instead of extinct a population of a wider distributed species. And that's quite important. So it's not only a curiosity about the names, it's just a very important thing for fishery assessment. And cryptid biodiversity occurs among other flying squids. Here there is the purple-back squid, that when people did study their DNA, they found different species. In this case, 5 of them. The thing is, there are 3 morphotypes. The dwarf morphotype here labeled as B. It's a very small one. Here the scale bar is 10 centimeters. Then we have in C the same scale bar that is 10 centimeters. This female is 5 kilograms in weight. And so we have the dwarf morphotype, the giant morphotype, and the medium morphotype. And what people found is that the dwarf morphotype and the giant morphotype were sister groups. That's something unexpected. And just to see how these animals look, here is a video of a sea wig, and here you can see the purple-back squid with the photophore here in this part of the body, in the dorsal mantle. That's a very interesting creature. So we have cryptid biodiversity that was correctly described in the genus Omastrephes, and here we have also cryptid biodiversity within the species Stenoteuthis wallenensis. Is frequent cryptid biodiversity among cephalopods? Yes, it is. In the case of the genus Ilex, there are 4 species, but it's impossible to allocate a male because the variation between the characters that allows you to identify each species overlap. So you can take apart from any species from them. We know that there are 4 species. They have their names, but it's almost impossible to identify it without using molecular methods. The same happened with Octopus vulgaris. It was a species that was considered to be, cosmopolite in the past, and now we know that there are several species with a smaller distribution range. The same that happened with Omastrephes, and the same happened with the Pharaoh cuttlefish. And currently I am working on the same topic using different families of oceanic squids, and we are finding descriptive biodiversity in many among them. So we are reaching the end of this talk. Thank you for bearing with me. And what we can say is that there were a lot of things that we didn't know about the flying squids before. In the last times, we have been studying a lot on their biology, relationships, and many other things. And even though these flying squids are among the most studied cephalopods, I truly believe that their amazing biology will give us plenty of surprises in the future. So I want to thank you all for your attention. This picture here in the left depicts my new kitten, that it was named after my favorite squid genus, Omastrephes. And that's what happens when my wife let me name a cat by myself. And also thank my funders. And if you want to reach and have a talk, you can find me in ResearchGate, Twitter, and also through my email. Thank you very much for your attention, and I will be happy to answer your questions. Thank you very much.